Speak and pause. Okay, so I think what's going on is that my kid put my tablet pen in his mouth and his saliva like shorted it out. So, and then I think he also unplugged the USB port to my Wi-Fi mic. Anyway, so I think I've got everything figured out. And I apologize for the technical difficulties. So what I'm going to do is I'm going to go ahead and start into lecture, into the chapter 16 lecture. We've already gone halfway through this, so I'm just going to continue with the lecture that we were talking about. So let me just close this and I'll minimize this. So we talked about Chargaff and we talked about Rosalind Franklin. We talked about Chargaff's rule and A's and T's and G's and C's paired. We talked about all this other stuff. And then this is sort of where we left off. So I just want to remind everyone that when we're talking about, let's switch this to red. So when we're talking about DNA replication inside humans, there's a few things that has to happen. So the first thing that has to happen is helicase has to find the origin of replication. So we shorten that to ORI. And remember, that's an AT-rich DNA sequence. And once it finds that origin of replication, it has to unwind or open it. So helicase finds the origin of replication, and it opens it. And I might ask you on the test, why is it A's and T's? And you should be able to tell me, well, it has only 2 hydrogen bonds instead of 3, which are G's and C's. So once helicase opens it up, we create a bubble, a replication bubble, and 2 forks. That bubble is here, and we cut it in half. You get a fork on this side, so this is a fork, and a fork on this side, which is the other fork. So that bubble is held open by what we call single-stranded binding proteins. I'm not going to write the whole thing out, but we already covered that. So these single-stranded binding proteins bind to here, and they keep them from re-annealing to each other because they're like magnets. They would go back together. So once that happens, the next thing that has to happen is that, remember, DNA polymerase can't do anything until it has, it needs double-stranded DNA, and it needs a free 3' OH group. Remember, there's an OH on this and a phosphate on the 5'. So in order to do that, primase has to come in, and it's an enzyme, and it puts down an RNA primer. And the purpose of this primer is twofold. So just like we said, it creates double-stranded. It's not DNA per se, but it creates that double-stranded condition, kind of like a train, so the polymerase can run on it. It can't. The polymerase cannot ride on here. These are the single-stranded binding proteins right there. Anyway, so it has to. The primer provides a double strand, both 2 tracks, for the polymerase to ride on, and so I guess we could call this A. And then B, it also provides that free 3' OH group, because it has to do those dehydration reactions that require the making of water. And you can't make water with a phosphate. So it can't, DNA can't get replicated in the 5' to 3 direction. I'm sorry, on the 5' end. It can only go on the 3' end. So it can only go from 5 to 3. It can only go this way because the 3 has a free OH. It can't go this way because the phosphate, we can't make water. So it cannot go that way, but it can go this way. And that's the only way it can go. And that's where we get this lagging strand where it's built in pieces and this leading strand where it's built continuously. And remember, we talked about the lagging strand is opposite on opposite sides and forks of the replication bubble. All right, so once the primer, and I'll ask you on the test, what's the primer made out of? So make sure you know it's RNA because it's really weird. RNA is real unstable. The next thing that happens is the DNA polymerase, and your body has several polymerases, but we're just going to talk about the bacteria ones, and those are represented by Roman numerals. So this specific polymerase is DNA polymerase 3, and you should know that. And DNA polymerase 3 is the polymerase that starts adding the DNA to the primed area. So here it's not showing the RNA primer, but trust me, it was there, and it was replaced with another polymerase. So DNA polymerase, which is shown here and here, 3, its job is it's the main copier of DNA. So it's the one that does most of the work. And so DNA polymerase will jump on there after there's a primer, and it will copy whatever the other strand is. So if there's an A on one strand, it'll put a T. And if there's a G on one strand, it'll put a C. And then the next thing that happens is you can't have RNA mixed in with DNA because RNA is unstable, and we learned about this in chapter 5. So we have to remove that RNA primer. So we're going to remove the RNA primer and replace it with DNA, which seems terribly inefficient. And this is done by a different DNA polymerase called DNA polymerase 1. And then on the lagging strand, remember that, and the lagging strand makes those pieces, and they're called Okazaki fragments after that dude that discovered them. On the lagging strand, these pieces right here, they can only be joined by DNA ligase. This enzyme can only join the 3' and 5' ends together. So there'd be another piece here, and this gap right here is the one that has to be put together by DNA ligase. So this is an important enzyme. Like if I wanted to take let's say that I wanted to make botulism toxin protein, and so I could inject it into people's faces and make $1 million. So what I do is I go to the botulism bacteria. I would take the gene for the botulism toxin out of it. I would put it in another bacteria that's not so toxic, that's easy to work with, like E. coli. And that E. coli, I would take a small piece of DNA, and then I would splice in to the E. coli's chromosome. I would splice in the gene from the botulism bacteria in order to force the E. coli to produce botulism toxin. And then I could extract that from the E. coli and purify it with a few steps. And if you guys take a biotech class or a genetics class, you could probably learn this. And then you just go and purify it and sell it, and and they inject it into people's faces to get rid of wrinkles, or it can also have other benefits. But anyway, I would, in order to link those pieces of DNA together, the piece from the botulism toxin and then the bacterial chromosome from E. coli, I would need DNA ligase because it's the only enzyme that could piece together those things. Think about it as like cutting and pasting, in a document. So ligase is what pastes the pieces together, and so it's required, but we only need it on the lagging strand, not the leading strand. All right, so that's DNA replication. And so there's an animation and a movie of this, and I'm gonna look this up on YouTube real quick. And I think that I also have the link. I should have the link under pages under the class. So if we go to Canvas and you go to pages, and then you go to web links, and the towards the end of the web links, it should have the video for DNA replication. So you can click on that.
The replication of DNA
and I'm going
begins at a sequence of new.
Play this, but I'm going to shoot. I need to share the screen. So okay. So this is directly from the links and Canvas under pages for DNA replication.
The replication of DNA begins at a sequence of nucleotides called the origin of replication. Helicase unwinds the double-stranded DNA helix, and single-strand binding proteins react with the single-stranded regions of the DNA and stabilize it. DNA polymerase 3 is the major enzyme involved in DNA replication. DNA polymerase 3 can only add a nucleotide to the 3' end of a pre-existing chain of nucleotides, and it cannot initiate a nucleotide chain. Therefore, an RNA polymerase, called a primase, constructs an RNA primer, a sequence of about 10 nucleotides complementary to the parent DNA. DNA polymerase 3 can then add deoxyribonucleotides to synthesize the new complementary strand of DNA. Because the two parent strands of DNA are antiparallel, they are oriented in opposite directions and must therefore be elongated by different mechanisms. The leading strand elongates toward the replication fork by adding nucleotides continuously to its growing 3' end. In contrast, the lagging strand, which elongates away from the replication fork, is synthesized discontinuously as a series of short segments called Okazaki fragments. When the DNA polymerase 3 reaches the RNA primer on the lagging strand, it is replaced by DNA polymerase 1, which removes the RNA and replaces it with DNA. DNA ligase then attaches and forms phosphodiester bonds. The DNA is further unwound, new primers are made, and DNA polymerase 3 jumps ahead to begin synthesizing another Okazaki fragment. For simplicity, DNA polymerase 3 has been depicted as separate units, one acting on the leading strand and the other acting on the lagging strand. The current view of DNA polymerase 3 is that the two subunits function together with the DNA on the lagging strand, folding to allow the dimeric DNA polymerase molecule to replicate both strands of the parental DNA duplex simultaneously. Proteins other than DNA polymerase 3 are not shown.
Okay, so
The replication.
So like it said at the end of this, it's actually
of DNA.
So it's actually it's done this way. So I'll just share this. This is a 3D model. It's kind of blurry because of the video quality, but you kind of get the idea. So
using computer animation based on the latest research, we are now able to see how DNA is actually copied in living cells.
So like it said, so one of these is looped out, and I don't know if you guys can sort of visualize this in your head, but if you had something that's 5:3, right, and you had a phosphate on it. So let's say you just glued a red sticker on the end of a thread and a white sticker on the other, and then you flipped it, so you made a loop out of it, then the top of that loop would be in the exact opposite orientation. So it would be like a 5 to 3 loop at the top of the loop. And that's what's going on here. So DNA is actually looped out on the lagging strand and copied backwards in the loop at the top of the loop, where the leading strand is continuously copied. So this is the leading strand, this is the lagging strand, this is helicase unwinding the DNA. And then these other things like primase and stuff are all connected together. So this is like one big giant conglomeration of enzymes. And generally we call that a holoenzyme. So that's what's going on here. And so I'll just go ahead and play the video.
You are looking at an assembly line of amazing miniature biochemical machines that are pulling apart the DNA double helix and cranking out a copy of each strand. The DNA to be copied enters the production line from bottom left. The whirling blue molecular machine is called helicase. It spins the DNA as fast as a jet engine as it unwinds the double helix into 2 strands. One strand is copied continuously and can be seen spooling off to the right. Things are not so simple for the other strand because it must be copied backwards. It is drawn out repeatedly in loops and copied one section at a time. The end result is 2 new DNA molecules. It's similar to copying information on a computer. Hundreds of megabytes of data can be backed up in minutes by burning a copy onto a CD. The copying process is accurate, typically less than 1 mistake in a billion. The DNA copying process in our bodies is even more accurate, 1,000 times better than most computers. Remarkable biochemical systems have evolved to double-check the new DNA strand and fix any mistakes.
Okay, so that's basically an actual simulation of how that works. And like I said, both of these are in under the pages if you just go to web links. And then we'll talk about transcription and translation and art and splicing and the last chapter, which is chapter 17. So there's more videos that you can watch. And then anyway, so I'm going to go back to sharing the PowerPoint. This URL doesn't work anymore. It is a good animation, but you have to pay for it. So it's not that good. All right, so if we look at the DNA when it's being replicated, we know that there's a problem with it going. It can only go 5 to 3 because of that that requirement for dehydration that we talked about in chapter 5. And so this is a parental strand, and this is the 3' end, and this is the 5' end. So we know way down here somewhere else that this has got to be a 5', and this is a 3'. And so these strands are going to come apart. Helicase is going to, separate them, and that's what this is showing. It's showing them being separated. So remember, the dark blue is the original strand, and then remember the RNA primer is put down by primase. So that's been laid down. What we're not showing is the helicase unwinding it and the single-stranded binding proteins holding it apart. But regardless, so that's what's going on. And then we have DNA polymerase 3, that's this light blue arrow. And we know that once it's primed once, DNA polymerase on the leading strand can continue to add on the 3' end because it has that hydroxyl group. So this can go on, and that's why it's called the leading strand. It only has to do it once. This is the lagging strand, and it has to prime it multiple times. So remember, it's going this. So this would be the first one, and then it would have to jump back and prime it and do this, and jump back and prime it and do it again. And that's why it's called the lagging strand. And each one of these things is Okazaki fragments. And this is linked together by DNA ligase, that enzyme that we talked about. And then remember the primers are removed. So anyway, when all that's said and done, there's a problem here. Whenever this primer here on the leading strand is removed by DNA polymerase, remember it's DNA polymerase 1 that removes that, there's a gap. And no polymerase in the world can add onto a 5' end because there's a phosphate there, not a hydroxyl group. And so that means that every time that your DNA is copied, one of the strands, the leading strand, gets shorter and shorter and shorter and shorter and shorter because it can't physically add all the way to the very end from that RNA primer. And so your chromosomes get shorter. And eventually it's gonna start. So let's say this is your chromosome, and here's an important gene right here. This gene is for glycolysis. And without it, you couldn't break down sugars to make ATP, and we all know you'd be dead. So eventually, maybe after 100 cell divisions, your chromosome would be this short, and then another 100 or so, it's starting to get into this gene. And right here, right, this gene would no longer work. So you'd have problems making ATP, and eventually the cell would die. So you get a couple hundred copies out of each cell line, but in the end, after that, they automatically undergo apoptosis. Now, there is a way to fix this, because I mean, you might think, okay, well, I came from a single cell, and a zygote that was in my mother's womb, and it divided, thousands of times. So how did I ever make it out to be born? And that's a great question. And the answer is DNA telomerase. So this is an enzyme that can add stuff to the end. So here's your chromosome, right? And DNA telomerase will just go on the end and add a bunch of junk to it, right? So even though, so let's say that that's the same thing, right? So DNA telomerase, I'll just do it on this side. DNA telomerase just adds a bunch of junk on the end of it. So you can't stop the chromosomes from getting shorter, but if you add a bunch of junk on the end, you can extend the life. So see, after this division, it's still not into this gene that's required. And so telomerase can extend the life, the number of divisions of a chromosome. And it does that by adding just junk DNA. So think about it this way. Let's say your driveway is 20 feet long and it's made out of gravel. And somebody every night comes and takes a foot away from your driveway. So after 20 nights, you wouldn't have a driveway anymore. But let's say that every night before then, you went out and put a foot extra on the end of your driveway. Well, even though you couldn't stop the person from taking a foot of your driveway, you'd always have a driveway because you're adding onto your driveway, even though they're taking it away. So that's what telomerase is doing. And remember, telo means end. So it's doing this to the end of the chromosomes. Telo is end, meros is part. So a telomere is the end part of a chromosome. And it's doing that it's just simply adding DNA at the end, and it's never going to be read into a protein. It's just a DNA sequence that prevents these cells from undergoing apoptosis or cell death. And that's how. So before you were born, telomerase is turned on in your body, but after you're born, it gets turned off. And there's a reason for that. So you wouldn't make it out of the womb if you didn't have telomerase. Because your cells would undergo cell death after a few hundred divisions. And even if I took a skin cell from you and I put it in a lab and a test tube, it would only undergo a couple hundred divisions, and then it would die. But here's the thing: if in cancer cells, they're immortal, And the way cancer becomes immortal is that it turns telomerase back on. If the gene stayed off and you had a cancer cell, cancer cells can't get around this problem. So they would only undergo a few hundred divisions and they would die too. So cancer really isn't dangerous unless it has this enzyme turned on. And you probably have it turned off when you're born as an evolutionary mechanism to prevent you from getting cancer. There are some people that have artificially tried to turn this on using CRISPR-Cas9, which is a gene editing system. I think they're crazy. I think they're going to end up getting cancer. But I guess, but the point is that they want to live forever. So this would make your cells immortal, but it also increases your risk of getting cancer because almost all cancer cells have this turned on. So I don't know. You might try to live forever, but eventually you're going to get cancer. So what would you rather die from, cancer or old age? Anyway, so telomerase, this is how telomerase works. It has a piece of RNA in it, which is shown here. Let's switch back to red. It has a small sequence of RNA that's attached to it. And this is protein, so the artist drew it as like a ball. And what it has the ability to add DNA. So it's a polymerase, but it's a special polymerase because it has its own primer. Its own primer is attached within it. So it fulfills the obligation of all polymerases. It has to be double-stranded and have that 3' hydroxyl group. And so it can do this all by itself. It can prime the DNA and extend it out, and it just extends it out so that it's much longer than the original DNA. So even though that gets cut off, it still doesn't cut into the original DNA. And so by doing that, it saves the genes that are what we call upstream or further in the DNA, closer to the centromere rather than the telomere. So this is the telomere, where telomere is end part, and then we have the centromere, which remember is the middle part that we learned about in chapter 12 and 13. And this slide is just showing you where the hotspot of activity is for telomerase, and as you guessed, it's on the end of the chromosomes. Yeah. So really, that's all I have much to say about telomerase. It's just basically adding junk sequences at the end. All right. So in that video, it talked about that polymerases, DNA polymerases, make about one mistake, 1 in 1 billion copies. And that's really good. I mean, if you think about it, it would be like you retyping a textbook that's the size, the height of the Washington Monument, and only making 3 mistakes the entire time. And of course, it would take you several lifetimes to be able to do that. And your body does that. Remember, you have 3 billion AGCs and Ts in you, and it takes, only a few hours to copy all of these. So it's really fast. It's super accurate. And the reason it can be this accurate, isn't because it doesn't make mistakes. It's because it has a whole bunch of proofreaders. So now imagine that you're typing, you're retyping the textbook, The Height of the Washington Monument, and you have a whole bunch of editors and proofreaders checking all your work before it gets sent off. And so by the end of it, it's not you that's never making the mistakes, it's just everybody else is catching your mistakes. And so you're only truly making 3 errors after all is said and done, instead of probably the tens of thousands of errors that you would make by actually typing this out. And DNA can do this because it has a proofreading mechanism, so it can read and check all the A's and G's and C's and T's to make sure that they're there. And if it does make a mistake, it can fix it. So the DNA polymerase checks for mistakes while it's adding the nucleotides. And it rarely makes a mistake, but it does, just like you type in a book. And it's up to other enzymes to catch that mistake and fix the DNA. And one of these enzymes is DNA polymerase 1, which we talked about, right? It's the same one that removes the RNA primer, and the DNA ligase, right? It's the one that can link the fragmented pieces of DNA back together again. So here's an example of DNA repair. And so what we talked about UV light, and you guys know by now that UV light causes you to get sunburned. You might not know what a sunburn is, but you know that if you stay out in the sun too long, it's going to happen. And what's really going on is that ultraviolet light can penetrate your skin. If you have dark skin, then the pigments get sucked up by the pigment sucks up the ultraviolet light before it gets to your DNA. So the darker your skin, the more you're protected from UV light. But you can also use sunscreen, same kind of deal. And regardless, the goal is to reduce the amount of UV. But if you're out in the sun long enough, no amount of dark skin or sunscreen is going to protect you from getting a sunburn from UV light. And what happens is that if you have two T's next to each other, so these are thymines, and they're in a chain, and so on the other strand we'd have A's. Well, these two T's, they link to each other. They get covalently bonded, and when it does that, it bends the DNA. Right? And so this is what we call a thymine dimer. Remember, di is two and merose is part. So a dimer just really means it has two parts. And so we know that there's two T's and a thymine dimer. And that causes this physical bulge because it's a covalent bond. It's a covalent link. And you guys know that covalent bonds are super strong. So nothing can undo this bond. And it has to be cut out. And what does that is an enzyme called nuclease. So DNA nuclease, it cuts here and here on each side of the damaged DNA, which is right here, the thymines, and it removes all of that. It removes it from this strain, only one of the strains. Now, DNA polymerase can get on here because it has the double strand, right? This is already acting as a primer, and it can add in nucleotides. So DNA polymerase 1 can fill in that gap, right? But it can't link these pieces together. Only DNA ligase can do that. So if you're out in the sun too long, well, if you're out in the sun at all, this is what's going on inside your body. These thymine dimers are being generated, and there's nothing you can do to stop it. As long as if UV light hits your DNA, it's going to cause this to happen. And when it does, it can't be undone very easily. It has to be cut out, and that it's cut out by a nuclease. So I'm going to ask you on the test, and then it's the missing strand is resynthesized by DNA polymerase, and it uses the A's on this strand to put in the new T's on this strand, and then ligase seals that nick. And so now we talked about the cell cycle, and you know that your DNA is checked at the G2 checkpoint. And so if you're out in the sun too long, you don't have enough workers, enough enzymes to fix all your DNA. And so what happens is that there's no way you're going to make it past the G2 checkpoint. And so the cells undergo apoptosis. And when they undergo apoptosis, they die. And your dead cells, they're inside your skin. They don't just fall off. Your white blood cells have to go in and phagocytose them. They have to eat them and digest them. And so that's what causes it to get pussy and red, because all of these white blood cells are coming out of your blood vessels to go and eat all the cells that died from being exposed to too much sunlight. which caused thymine dimers that there weren't enough workers to fix. You probably didn't know that. Maybe you didn't want to know that much, but anyway, that's how it works. And that's the end of chapter 16. I normally would ask if you have any questions, but nobody's actually there. So what I'm going to do is I will cover this in a review, and I'm just going to let you guys know. So the plan is I'm going to lecture a little bit on chapter 17, maybe another 10 or 15 minutes. Then on Thursday, I should be able to finish chapter 17. Next week, we will have our final lecture on chapter 17 on the 18th, and then a review, and then your exam will be open on the 19th. And then I'll have the grades done. And if future classes are watching this, this is for spring, specifically spring of 2021. So anyway, just letting you guys know the semester is wrapping up, and so we just have one more class, and that's sort of the game plan. All right, so I'm going to stop sharing this, and then I'm going to go ahead and open the next lecture, chapter 17. Okay, so chapter 17 is how we go from a gene to becoming a protein. And you guys remember the central dogma, so that's kind of what we hammered at through the whole semester, is that DNA is turned into RNA, and RNA is turned into protein, right? And it's this protein that makes us what we are. This makes us flesh and blood. Remember, DNA is found in the nucleus. There's only one copy. It never leaves the nucleus. RNA is a messenger. It sort of takes the message from the nucleus to the cytoplasm. And remember, it's the ribosomes that we talked about in chapter 6 that convert the RNA. They read the RNA and make it into protein. So we're going to talk about how this works. So anyway, when you go from DNA to RNA, you're going from a 4-letter code to another 4-letter code, right? And so these are very similar languages, and so we call that transcribing if the language is similar. So from going from DNA to RNA is called transcription. It's kind of like what a court reporter would do. And then when we go from RNA to protein, this is a 4-letter language, and this is a 20-letter language. And so going from a 4-letter to a 20-letter language is way different. It would be like going from Greek to Latin. And you guys know that if you did that, you would need to translate the one language into another. So this process from going from RNA to a protein is called translation. And they're the two main processes that allow this central dogma that allows DNA to be made into protein. You can think of DNA as a cookbook and proteins as the food that you eat. So you've got to remember that these are two separate events, and it's very common for students to cross these up or mix them up. You've got to have them on different plates, right? So you've got to have transcription in one pile while we're going through this, and translation in another, because they're completely different steps. All right, so we're going to start sort of defining this. So transcription is done, it converts DNA into RNA. That's done by an enzyme called a polymerase. All these that make polymers from monomers are called polymerases generally. This one is specifically an RNA polymerase. And if you talk to a molecular biologist, they would call this a DNA dependent RNA polymerase. I'm sorry, my handwriting is terrible. But it's a DNA dependent RNA polymerase. And that means that it relies on DNA as a template, as its original copy, to make an RNA copy of it, right? So it's opening the DNA and it's reading the DNA and making RNA, which is what's going on here. Remember, this is making single-stranded RNA from double-stranded DNA. So it could use either strand of DNA. We're just going to, but it had, just like every polymerase, it can only go from 5 to 3. So no matter, so if this is the RNA that's being made in a 5 to 3 manner, then the DNA itself, the original DNA, it has to be on the 3 to 5 strand, not the 5 to 3 strand. So this would be the double-stranded DNA, but it would only be able to read this strand in this direction. It could read this strand, but it would have to go in the opposite direction. And usually we don't write it that way. All right, so that's what transcription is. It's taking double-stranded DNA, reading one of the strands, and just converting it into the letters of RNA. So for example, if it's reading this strand, so let's say that this is an A and a G and a C and a T. Well, the RNA polymerase would say, aha, well, that's an A. I'd like to put a T in here, but I'm an RNA polymerase, so I can only make RNA. So I have to use U's instead. And then the next letter it sees is going to be a G. And so it was going to put a C in there. This is a C, so it'll put a G in to match it. Those are 3 hydrogen bonds. These are 2. And then a T is there, so it'll put an A in, and so on. And so this would be the RNA strand, and we call that messenger RNA or mRNA. All right. So there's a lot of stuff that has to happen before this mRNA leaves the nucleus, and we call it RNA processing. We'll talk about that in a second. All right, so on to translation. Translation is taking this messenger RNA, and a ribosome will actually look at the letters in here, and it will read it and turn it into what we call a polypeptide, which is a fancy name for a protein. Same thing. So ribosomes are going to read this mRNA, and it's going to turn it into that 20 letters amino acid that we talked about in chapter 5, where we talked about how it folds into its correct shape, and that's really important. So just remember, ribosomes do that. Now, eukaryotes and prokaryotes do this differently. In eukaryotes, we have nucleus, right? So all of the transcription is done inside the nucleus, inside the nuclear envelope. In prokaryotes, they don't have a nucleus. So the mRNA is made in the same place that the ribosomes reside. This is 2 separate locations. Ribosomes do never go inside the nucleus. And DNA never leaves the nucleus. It's never in the cytoplasm. That's very different in prokaryotes. All right. So when we're looking at an actual gene, the gene itself is made of 3 segments. So let's just say that this is chromosome number 1. And this is from a human. And we'll just say on here is a gene for, let's just say it's hair color. There's a few genes involved in hair color, but let's just say it's hair color. And let's say in this case, it's brown. It's for brown hair. And let's say that you got this chromosome from your mom and you know you have one from your dad. And let's just say dad's is brown too. So there's 2 chromosome number 1s. And so we're going to zoom in on this gene, which is at a specific location. So remember, we call that the locus. It's short for location. So that gene would be in a 3 to 5 prime orientation. So this would be one strand of DNA. This would be the other. This would be 3 to 5. This would be 5 to 3. And so there would be some letters in here like A, G, C, and T, and so on. That would be converted to mRNA, and then that would leave the nucleus and then go into the cytoplasm C, where the ribosomes would find it, and then they would read what's in the mRNA and they would make it into a protein, kind of like you reading a cookbook and taking the ingredients and making, I don't know, shepherd's pie or whatever you like to make. So the question is, what constitutes a gene? What are the parts that make this brown-haired gene? And for every gene, there's always 3 parts. One part is called the promoter. The promoter is kind of like an on-off switch, but in humans, it's like more like a dimmer switch. So a promoter is responsible for regulating whether or not a gene's on or off, and how much it's on or off. Like a little on, a lot on, and so on. So that's the promoter part. The other part is the coding region. So the coding region, so this would be the promoter. This would be the coding region. And this would be the actual part that the ribosomes are going to read and turn into a protein. So the coding region is actually turned into amino acids. The promoter isn't actually read by the ribosomes because it's never made into RNA. The RNA only actually starts right here, where the coding region starts. And then we have a terminator. The terminator sequence is right here at the end. And its job is to stop the transcription of the gene so that it has a specific stopping point. So if we looked at a gene, it would have a promoter, coding region, and a terminator. And all these are sequences. All right. And every gene has this. Okay. So if we look at the promoter, if we're just going to go back and just look specifically at the promoter itself. So we have the promoter and the coding region and the terminator. If we just looked at this promoter, and we're just going to zoom in on the promoter. So the promoter, these are all promoters in different genes. And we can talk about this gene. So you guys know the beta subunit of hemoglobin. Remember we talked about this in chapter 5. So there's alpha and beta subunits, and 2 alphas and 2 betas make up a functional hemoglobin. And if you don't remember this, you can go back to chapter 5 and take a look at it. And you'll see there's an actual picture that I have the 4 subunits that make hemoglobin. So this is the beta subunit. And these are the elements that are found in the promoter. So there's like specific sequences in the DNA. So this is called a cat box because it has these letters in it. This is called a Tata box because it has these letters in it, right? This is a virus, simian virus promoter, and you can see it has a lot of these GC sequences in it. And so the reason I'm showing you all this stuff is I don't want you guys to memorize all this, but what I want you to see is that it's kind of like a barcode, like a UPC symbol. You can see that the order of these are different, and it's going to tell the genes to be on or off or on at a specific level depending on the need of it. So you need hemoglobin all the time, but only in specific cells, only in red blood cells. A virus job is to make as many copies as it can. So this is going to be like screaming on because it wants to make as many copies of viruses as it can and get out undetected. Kinases, we talked about these, right? You need these to add phosphates. And then histones, this is the subunit of one of the histone proteins. So you only need these when you're copying DNA. All right, so the promoters, in the end, the promoters have different sequences. Those sequences, the order that they're found in that promoter region is important. This is where the coding region starts. And we number that as like +1 for the first nucleotide of RNA that's made from the gene and DNA. So if this or the promoter here, this is the coding region, and this is the divider, this would be +1, whatever this, if let's say that's an A, and if this is a C right in front of it, that would be -1. This is never made into RNA, only the plus numbers are made into RNA. All right, so this is how transcription works. We have the promoter, we talked about it. This egg right here is the RNA polymerase. The artist decided to make it an egg. This is the start point, so remember that would be the +1. And then this would be the right behind it would be -1. This the RNA polymerase goes and finds the promoter and opens it. And remember, it can only copy 5 to 3. So it's only going to be able to copy this strand. It cannot copy this top strand. That's why it's starting here, right? So if that's 3, this is 5, and it's going to continue to copy it in a 5' to 3' direction, and this pink is going to be the RNA transcript. And then the polymerase keeps going until it hits this terminate. It's like a physical U shape. I'm not going to test you on this, but imagine if you're a train running down the tracks and there's a big giant steel bar across there. Well, it's probably going to make your train fall off the track. So these structures that can be produced, remember, DNA is like a string, like a shoelace. It's not like a stiff piece of rebar. So it can make weird shapes and structures, and it can fold and bend, and do all this stuff. And so it can make these weird shapes that cause the polymerase to fall off the track, so to speak. And so that's what happens. So when it hits this terminator, it falls off, and we get the completed RNA transcript. So this is only a copy of the coding region, right? So the promoter turns the gene on and off. This is what stops it. But the RNA is actually just the coding region of the gene itself. There are 3 steps to transcription: initiation, elongation, and termination. And so initiation is the start of transcription, elongation is the continuation of transcription, and then termination is the stop of transcription. And that's just because scientists love to classify stuff. All right, so we're just going to talk about initiation first. There are proteins called transcription factors. Those transcription factors recognize these DNA sequences. So there's a thing called the TATA binding protein. I don't care that you know this, but that's what this thing is. So a TATA binding protein is a transcription factor. And so what it does is it recognizes and binds to the TATA box sequence. So remember, that's the TATAAAT sequence. And so that transcription factor binds there. And that it's kind of like a person flagging an airplane to park in its spot. So once that transcription factor binds, it's going to recruit all of the other stuff, including the RNA polymerase. You don't need to know that this is specifically a 2, but we distinguish them by what they do. So the RNA polymerase will recognize all these other transcription factors that are going to also bind to the other sequences that they recognize, and it's going to park like a plane at the right gate. And when it does that, that's what we call the transcription initiation complex. It's all these transcription factors coming together to recruit the RNA polymerase. If you want to think of it, the RNA polymerase as a plane, then these transcription factors might be all the workers that are trying to get it to go to the right gate. You know, the ground crew, the air traffic control, that sort of junk. Once it finds the right place, it's going to open it up and it's going to start copying the DNA and turning it into RNA. Elongation is not really that exciting. About 20 nucleotides get opened up at a time. You don't need single-stranded binding proteins because it's very short. It's not like all the big stuff going on with DNA replication. And remember, it has to go 5 to 3. So if it sees a T on the DNA strand, and this would be the 3. To kind of follow this, this would be the 3 to 5 strand, and then so the RNA would be 5 to 3. And if it saw a T on the 3 to 5 strand, so let's just draw it out. It sees a T here, then the RNA is going to put in an A, and then if it sees an A, then it'll put in a U, and so on and so forth. And it just continues to do that until, you guessed it, it hits the terminator, and that terminator causes it to fall off. And there's a video of this in that pages of Canvas as well. All right, so we've got our DNA copied into RNA, and so we call this pre-mRNA. The reason it's pre-mRNA is because you've just basically copied the coding segment of the gene, right? Remember, we have the promoter, there's an M in there, right? Promoter, and then the coding region, and then the terminator. And so we're just copying the coding region basically into RNA. So there's some modifications that go on both ends of this RNA and then within the RNA itself. The first thing that happens is on the 5' end of the RNA, we add a G cap. This helps protect the RNA from being degraded by enzymes, because the cytoplasm is full of really mean enzymes that want to chop up RNA. But it also helps the ribosome recognize which direction. So let's just pretend like the RNA was made into this word rat, right? And this is the 5' and this is the 3'. Well, when the polymerase comes on here, the polymerase is going to come on here, and it's going to read this in a 3-letter code. We'll talk about that why. And then it's going to turn it into, you know, let's say it turns it into an actual rat. Well, if the ribosome read it in the opposite direction, the direction it's not supposed to read it in, it wouldn't make rat, it would make tar instead. So it's super important that the ribosome only starts reading on the 5' end, and the GCAP helps to tell it, hey, this is the right end to start reading the RNA and turning it into proteins. So that's the reason for the 5' cap, and this is what it looks like. You just need to know it's a 5' cap, and you need to know what its purpose is. You should also know it's made out of it's a G. Okay. Now so we've got our RNA. I'll make it green. So we've got our RNA. Well, that's red. Whatever. I'll just make it red. And so remember, it's 5 to 3. It's been made that way. Now we put a G on the 5' end, and that's to help the ribosome and to help protect it from getting chopped up by mean enzymes. This is all still in the nucleus. So it's still got to go out to that harsh environment of the cytoplasm, which is real scary, like going into the woods. And so we also need to protect the other end. To do this, we add a whole bunch of A's on it. And this is the most vulnerable part of the RNA. So this is the messenger RNA. Is this 3' end. So we can vary the tail. Think about it as the telomerase, right? So when you get in the cytoplasm, these RNases, these mean, nasty enzymes, are going to start cutting this up. They're called endonucleases, and the reason they're called that is because they can only chop. They're called exonuclease. They're called exonucleases because, and I just figured that out from this, but they're called exonucleases because they can only cut from the end. They can only cut from this end, and they can only cut from this end. And so these enzymes are going to find this RNA, and they're going to try to destroy it. And so if you had a really long segment of A's, and you had just like a couple, that enzyme is going to cut at a specified rate. So if it finds this mRNA, it's not going to take it long to start cutting into the coding region and destroying that mRNA. But if it's like 100 A's on the end, it takes it a lot longer to physically chop all of these A's off before it actually gets into the coding region. So the longer the poly A tail of this mRNA, the longer it lasts in the cytoplasm, which means the more ribosomes can read it and turn it into a protein. So the more, kind of think about it as like if you wrote down a recipe and it had an expiration life where it just kind of burst into flames. So you could make a lot of cookies before it actually burst into flames if it was an hour versus a minute, if that makes sense. So the modification of the tail helps inhibit degradation. We talked about that. It also helps tell the ribosome, don't start reading on this end, only read on the other end, on the 5' end. And it also might help regulate synthesis of proteins because the mRNA is not allowed to ever leave the nucleus until this tail is put on it. And we know that because we've chopped off the tail and the mRNA can't leave the nucleus. So those are the reasons for the poly A tail. And you want these mean enzymes like RNases to chop up your RNA in the cytoplasm, because we talked about this, like if you went outside and you were cold, you might start shivering, but when you get warm, you want to stop shivering. So if your mRNA that made you shiver was around forever, you would never stop shivering. So we talked about earlier in the semester about carbon 14, and that it has a half-life of thousands of years, or 5,700 years. RNA, so this is years. RNA also has a half-life. We call it a half-life, and that's how long half of the RNA would stick around and get chopped up by the other half, would get chopped up by enzymes. And so this is usually in minutes rather than hours or years like we would measure radiation. And you also measure half-life of drugs. So let's say you took ibuprofen, the half-life would be how long it takes for half of it to get degraded by your liver. So it's the same sort of deal. All right, so we got the coding region that we turned into RNA. We've got the cap, and we've got the tail. But we're not done yet, because we need to edit the inside of this. And that's called RNA splicing. So RNA splicing is kind of weird. And so what happens is so this is a pre-mRNA, 5' cap, remember that's a G, poly A tail. This can vary depending on how long you want this RNA to hang out inside and get turned into protein. But we also have these things called exons and things called introns. And I know that this is a foreign language, so you're going to have to probably just memorize this, like if you were learning a Spanish class or something. But exons are differentiated by introns because introns get cut out. And the exons get left, they get linked together. So we could call this exon 1 and exon 2 and exon 3. So we were looking at this, the introns would be gone, and this would just be exon 1 and exon 2 and exon 3 all linked together. So the introns are excised and the exons are linked together. We call it spliced together. So you're basically cutting out the introns and tossing them into the lysosome and getting rid of them. You're chopping them up and they get made into another RNA. This seems highly inefficient, right? I mean, why bother making this to begin with? But there's a purpose for this. And the purpose is that you can make a lot of different proteins from a single gene by doing what we call exon shuffling. I'm going to change to red because this is easier to see. All right, so let's simplify this. We have exon 1 and we have exon 2. And you guys know that introns are going to get chopped out. So there's an enzyme that comes in. It's called the spliceosome. It has these small nuclear RNA particles in it and other proteins. And basically what it's doing is it's doing a hydrolysis reaction, which we talked about in chapter 5. And it's cutting here and it's cutting here. And then once it's cut, then it does a dehydration reaction where it takes these two pieces and links them together. So in the end, you get exon 1 and exon 2 linked together, and then that intron is excised and it goes on to the lysosome where it gets chopped up into nucleotides, the individual A, G's, and C's and U's, because it's RNA. And then those get recycled, so they get made into a different mRNA. All right, so what's the purpose of that? Well, the purpose of that is that you can do some interesting things. So let's just back up a little bit. We have the promoter, and we have the coding region, and we have the terminator, right? And that's what makes a gene. And this coding region is what's eventually going to be made into an RNA, right? This is a pre-mRNA. And we're going to do some editing to this. So we're going to add a G cap. So that's the 3' cap. And we're going to add a poly A tail. Sorry, this is the 5' cap. This is the 3' tail. So we're going to add a bunch of A's to the tail. And now it's no, and then we have to cut out the introns and the exons. So we're going to, so let's say this is exon 1, this is exon 2. So we're going to cut out the introns. And in the end, we're going to end up with just those 2 exons linked together. And that's going to be what we call the final mRNA. And that's going to go out of the nucleus into the cytoplasm C to be turned into a protein by the ribosomes. Now, if we cut out these introns, then we can reshuffle these into any order we want. So think about it this way. You guys probably seen those magnets where they have different words on it, like the, and then, you know, maybe rat and, you know, 8, and then maybe it says cheese, and then you got another one that says, you know, cat, and so on. And so you can think about these as exons. So let's say that this is exon 1, and this is exon 2, and exon 3, and exon 4, and exon 5. So that sentence would say the rat ate cheese cat. All right, so that's a sentence. I mean, that's not a very good sentence, but, you know, let's say it was written by a kindergartner. So, but we can change the sentence, right? If this is one continuous piece, and it said the rat ate cheese cat, you could never rearrange these words to make a new sentence. But because we can get rid of these introns, then they all become separate words. Every exon is a separate word, and we can shuffle them. So we could say the cheese ate. Wait, let's do another one. So one that makes more sense. Let's say the rat ate cheese, and we could just get rid of that. We don't need that one. So we can make that sentence, or we could say the cat ate the rat, or the cat ate rat. So that's a different sentence, right? Or we could just say the rat cheese, or the cheese ate, or whatever. We can make any sentence we want, and hopefully you get the idea, because we can link exon 1 plus exon 3 plus exon 2, or we could start with exon 3 and link that to exon 2, and then link that to exon 1. And so this is called exon shuffling. Or alternative splicing. And that allows us to take one gene and rearrange the coding region of it to make multiple mRNAs so that we can make multiple proteins. And those are usually our domains. So domains are like areas where maybe this recognizes and binds to DNA. And maybe this is the active site that we talked about in chapter 8. And maybe this is a transcription factor that turns DNA on or off. So you could have a protein that have multiple areas of it that do different things, and that's what we call domains. All right, so I'm going to stop here. We've covered transcription, and so on Thursday we'll cover translation. And I would bet that I'm probably going to finish chapter 17 on Thursday, and then we'll do a review on the following Tuesday, and then you guys will take your final exam. And then we'll wrap up with grades, and we'll be done. All right. So again, I apologize for the technical glitches and snafus, and I will post this as soon as I can on YouTube and then link it in Canvas. All right, have a good night.